membrane begins to disintegrate, and at the end of prophase, the centrioles are at opposite poles and the membrane has disappeared. The double chromosomes attach themselves to spindle fibers and line up at the middle of the cell. In anaphase, the centromeres split. Half the chromosomes move to one pole, half move to the other pole, and the cell membrane begins to pinch in the center. In telophase, the nuclear membranes form around separated chromosomes and form two cells with the same structures and number of chromosomes as the parent cell. Homologous chromosomes pair to form bivalents. The centrioles divide and move to opposite poles of the cell. The bivalents duplicate to form tetrads or form chromatid groups. The nuclear membrane disintegrates, crossing over occurs. In metaphase 1, the tetrads attach to spindle fibers at the centromeres and line up at the middle of the cell. In anaphase 1, the tetrads separate and the pair of chromatids move along the spindle to their respective centrioles, and the cell membrane begins to constrict. In telophase 1, the nuclear membranes enclose the separated chromatids, and the cell membrane completes its constriction to form two cells, each with the same number of chromatids as the parent cell. In prophase 2, the homologous chromosomes do not duplicate, but they merely separate. In metaphase 2, the chromatids line up at the middle of the cell. The centrioles and asters are at the poles a spindle has formed. In anaphase 2, the now separated chromatids approach their respective poles. The cell membrane begins to constrict. In telophase 2, there are now 4 cells, each with half the number of chromosomes as the parent cell.